So I'd like to just finish today by telling you a little bit about the NSF EPSCoR project, which is titled Building an Intra-Organ Vascular Tree. We call this a vascular tree because we're interested not only in forming a vasculature to nourish bioengineered organs, but we want it to be hierarchically branched and have a hierarchical series of vessel diameters. So we want to start with a trunk that would feed the organ. So it would be like the main artery that comes off of your aorta to feed your kidney. But when that goes into the kidney, we don't want that same amount of blood volume and pressure to hit cells, so we want it to branch. So you have the tree branches into minor branches, into twigs, and then into the little veins in the leaves. So an approach that we're using to generate this vascular tree is to use these tissue spheroids that you heard about earlier today, and those can be homo cells, meaning one type of cell, or they can be heterocellular spheroids, meaning composed of a variety of cells. And the theory behind this, as we spoke about this morning, is that if you can print these spheroids into a vessel configuration, they naturally undergo this fusion process to form a tube. And we show that with real cells up here, where you can see that two spheroids, which are placed at close apposition, over time will fuse. It's a natural biological phenomenon. So our idea, and you saw this slide earlier today, is that initially, if we can use these cell aggregates and place them in a number of configurations, we can theoretically print a branching vascular tree. And due to cell aggregation, fluid dynamics, we actually have these spheroids form a lumen at their center and fuse into a tube. And then as we move ahead with the technology, we can use these sort of three-dimensional constructs with a number of cell types to actually print the organ as well as the vasculature within it. And as these cells fuse, like cells with like cells, you form kidney tissue, which has a built-in intra-organ vascular tree. So, and South Carolina leads the nation in this technology. Bioprinting and the cell aggregation application to bioprinting was developed here at Clemson University, the Medical University of South Carolina, as well as USC. So this is really our chance to excel and lead the field in biofabrication for regenerative medicine. So thank you very much.